All right, good morning. Let's start our class and we'll start by going over the warm-up. So let me share the screen here. And let's take a look. All right, so which of the following is true when light hits a rod photoreceptor cell? So this week talked about the whole process of phototransduction. Again, that it all starts with this. Cisretinal converts to transretinal. And then again, it's gonna detach from the opsin. The activated opsin is going to activate that G protein, the transducin, which is gonna start a second messenger pathway, which turns on phosphodiesterase. And when phosphodiesterase turns on, it breaks down cyclic GMP. So that's the next one. Cyclic GMP levels go down. When cyclic GMP levels go down, then that closes the sodium channels. When sodium conductance stops, the cell membrane voltage goes down. And again, that is the actual electrical change. That's the transduction there, from light to an electrical change. So, and again, most people got that. Semicircular canals, almost everyone got this. Head rotation, 3 different axes. What is the one that tells you the direction of gravity?
Saccule and utricle.
Exactly, the saccule and the utricle, also called the sacculus and utriculus. The actual sensory transduction in hearing is done by, the hair cells. We kind of talked about the hair cells ad nauseum as the key mechanotransducers in all the different senses within the ear. How is the outside? So this, I haven't graded all of these, so that's, but the idea that middle ear is filled with air, and unless you equalize the pressure with the outside, your eardrum's going to implode or explode. So again, most people were getting it, the Eustachian tube, the little connection between your middle ear and the back of your throat. It's also called the auditory tube or the pharyngotympanic tube. Those are all synonyms. And particularly like when you yawn or something or swallow, it kind of pulls open and lets the air pressure equalize inside and outside. Again, if you've got a cold or for allergies or something that inflames that, it can make it trickier to equalize the pressure. And then finally, photoreceptors responsible for high visual acuity. That was the cones. Most people got that. The ones that do color is just the cones. The rods are purely monochromatic. They can tell presence or absence of light. Convert electrical light into electrical signals. That's both of them. They're both phototransductors. Center of the retina, that's the cones. The rods are on the periphery. And finally, at least everybody got, this is the one everybody got. Only rods are the ones that can function in very dim light. The cones need bright light in order to register a signal. So speaking of rods and cones, one person did actually notice this because they did the assignment, but most people didn't. So I want to make sure everybody realizes I added a very simple little do-at-home assignment for people to get a feel for rods and cones. It's called First-Hand Experience of Rods and Cones. It's here in the module for this week, which I need to rename. This is not cardiovascular system. Let's just change that right now. This was finished nervous system. So firsthand experience of rods and cones is basically just saying, for you to do, and this is only going to take you a few minutes. I talked about the differences between rods and cones, but I really want you to experience it. Normally we do it together in class, but we can't, obviously. So this is something I want you to particularly pay attention to: color, whether things are colored or not colored. I want you to pay particular attention to visual acuity. Like, have a piece of paper with some writing on it in your hand so you can look at it and see if you can read the words or not. And then it says, turn the lights on so it's nice and bright. You're using your cones, and you're going to pay attention to your experience of color and your visual acuity. Can you read the text on the paper in front of you? Second, turn down the lights way down low. Maybe it's going into a closet. Maybe it's waiting till nighttime and turning off all the lights. Figure out the best way for you to do it. There's got to be a little bit of light, but really not much. And then describe what you see immediately, which is probably going to be very little because your cones are no longer working, but the rods haven't had a chance to do their dark adaptation yet. So what does it look like when you first turn off the lights? And then 5 minutes later, what does it look like? And things stabilize with the cones out of the picture, but the rod's now working. So rod vision. Now, what does color look like? Do you see colors or not? How's your visual acuity? Look at the paper in front of you. Can you make out the letters or not? So, and take note of that. And then turn the lights back on and see how things shift. So you have this instant, you can't have an instant comparison from cones to rods because it takes 5 minutes for the rods to come online. But you can get a pretty instant, once you're used to like, oh, this is rods, turn the lights back on. And it's like, whoa, let's see how everything looks different. And I just have a little text box. I just want you to type a few sentences in about what this experience was. So I just want to see that you actually have this kind of visceral experience of, oh yeah, that is what cones are doing. Oh yeah, that is what rods are doing. And just type it in the little text box and hit submit and you're done. So in this, again, it's just a little homework thing within the module for this week, which is finish nervous system and do muscular system. So sometime before the exam, you should do this. And it's going to be part of your kind of studying for the exam, right? Because it's also re-emphasizing the properties of rods versus cones.
Professor Eager?
Question?
Yes. Could you quickly go over the relationship between bleaching, like after image, negative image, and dark adaptation? I'm just kind of getting confused when I use which terms.
So the bleaching actually happens because, so like that negative after image is due to bleaching.
Okay.
Right? The cones don't bleach in the same way that the rods do. The rods bleach and they take 5 minutes to come back. When the cones get like kind of overexcited, they're back online in a moment. When you saw that bee, the bee looked, you saw the color in the bee for a moment, but then all of a sudden it was black and white again. Mm-hmm. But it's the same basic idea. When the cones have been working and working, then they also kind of take a moment to recover. But instead of taking 5 minutes, they take a fraction of a second there.
Okay, thank you.
The other part, the positive after image, comes from kind of them being excited and not shutting up right away. Right. So that's another, there's two things going on. One is a positive after image where they, sometimes if you like look at a bright light and you keep seeing a bright light, that's when they don't shut up. But then all of a sudden you start seeing the opposite color is when you actually start having those receptors aren't talking and you're getting the negative after image of what you're seeing minus what they would have been adding in.
So is the positive after image that kind of continued chemical reaction of the cones? Or
Yeah.
is that, okay.
Yeah. Mm-hmm. All right, so we are gonna have to really, we have a lot to talk about muscles. So I'm gonna go into the muscular system. It's not, I mean, what do you call it, a system? It's more muscles. So what is kind of a defining quality of muscles? Like if you were going to describe muscle tissue, what do muscles do? What do they do?
They contract.
So they contract. So all muscles contract. In fact, they only contract. We're going to talk about how you, because obviously using your whole musculoskeletal system, you can push, you know, so, but muscles can't push. Muscles only contract. We'll talk about just the mechanics of how we do that. They're excitable. We are going to see that they have the exact same voltage gated channels in their cell membrane that we find in the axon of a neuron. So we're going to have the same chain reaction action potentials running across the muscle cells that we saw running along the axon of a neuron. So they only contract and they're excitable. So that's going to be kind of a common theme no matter what kind of muscles we're looking at. We'll see different kinds of muscles. What are the three main kinds of muscle tissues that we get?
Skeletal, cardiac, and smooth.
Yeah, so there's skeletal muscle. This one is voluntary. Sometimes this is what we think of as being activated by the somatic motor system. This is the ones like your biceps and your triceps and all the muscles you use to run and kick a ball or whatever. It's often called striated. Striated means striped because under the microscope, you can see these little marks across it where it looks kind of striped. And we'll talk about why that is the case. It has to do with the arrangement of the proteins, the contractile proteins that make it up. They are laid out in this linear alternating arrangement. So under the microscope, it looks kind of like little hatch marks across it. So skeletal muscle, this is where we're going to spend most of our time today. We're going to talk about the details of how skeletal muscle cells are put together, how they generate their contractile force, different ways they have to make ATP to power themselves. Different versions of skeletal muscle fibers are optimized for endurance or for explosive bursts of strong energy. So this is where we're going to spend most of our time, is the skeletal muscle. Again, you've already been introduced to it when we did the somatic motor system. You got to meet the neuromuscular junction with its little nicotinic cholinergic receptors binding acetylcholine released by the somatic motor neurons. That's these dudes. What other kind of muscle tissue do we got?
Smooth.
They're smooth. This tends to be involuntary. You know, this is the kind of muscle that is in the walls of your stomach squishing up your food. This is the muscle that's in the walls of your blood vessels, adjusting the diameters of your blood vessels. This is the muscle in your eyeball that's adjusting your iris and the focus on your lens. This is the muscle that's being activated by the autonomic nervous system, the sympathetic and parasympathetic motor fibers. And as we're going to see, it can also get activated by non-neural stimuli. Skeletal muscle is always activated by a somatic motor neuron. Smooth muscle, on the other hand, has lots of different ways you can activate it. You've already seen, like in sympathetic, you can use adrenaline, epinephrine, just a hormone can activate smooth muscle and, dilate your bronchial passages or change the diameter of your pupils or something, right? So smooth muscle can be activated by neurons, but it can also be activated by hormones, and we'll see by other chemical signals and even stretch and things like that. So we will talk about smooth muscle, not in as much detail, but we'll definitely talk about it in the ways specifically that it contrasts with skeletal muscle. And then finally, what other kind of muscle tissue is there?
Cardiac.
There's cardiac, which you find in the walls of the heart. We will talk about cardiac muscle, but we'll wait to talk about that in detail till we get to the cardiovascular system. It has its own specialized properties that optimize it for, being the muscle that pumps your blood. So what we're going to do now is we're going to start with this. We are going to, before today is done, we will also be spending some time talking about the smooth muscle, but we are going to start by talking about skeletal muscle. And when in your book, when it talks about the muscular system, they're usually talking mainly about skeletal muscles. And I should mention, obviously they're about movement. They're moving your skeleton around. What other functions do they play? They do more than just for movement.
Protection.
So definitely protection. If you think about all of your abdominal organs, like your stomach and your intestines and all that, they are basically totally exposed to the outside world except for layers of abdominal muscles in front of them. We've got, actually, if you've been in anatomy, you've got like those 4 layers, the transverse abdominis and internal obliques and external obliques and rectus abdominis. You've got, it's basically like a kind of plywood with fibers in different directions, making a nice wall to protect all your internal organs. But those are the things that are actually in front of all those organs. It's not like your skull protecting your brain or your rib cage protecting your ribs and your heart. For all your abdominal viscera, they basically got some skeletal muscle. What other functions?
Stability.
So so it's kind of stability. A lot of your joints are stabilized just by the muscles around them. So kind of stabilized joints, also just your posture, right? Muscles aren't necessarily moving, but they're keeping you from slumping over. Plus muscles are what are keeping your shoulder joint together. The head of the humerus really doesn't have much of a connection to your shoulder blade, except for getting pulled in by the muscles of the rotator cuff. Your knee joint is also really kind of not much holding it together besides the pull of the tendons and all that. There are ligaments and things, but the muscles are are major players in stabilizing your joints. Like if you sprain something or dislocate something, one of the main ways you can stabilize a joint is just by strengthening the muscles that are associated with that joint, so they pull things together and hold the bones together better. So that's another big role the muscles are playing here. What else do muscles do for you?
Generate heat.
Definitely. So temperature regulation. So a muscle is basically a motor, right? It is something where you give it some kind of fuel, in this case, ATP, and it moves, right? You put gas in your car and your car rolls along, or you put wood in the steam engine and steam engine starts moving or whatever. Muscles are these things, you feed them ATP and they move. Again, if you've been in physics, you know that engines are always inefficient. There's never a complete conversion of the energy in the fuel into kind of movement and force by the engine or the motor. So in your muscles, large part of the energy that is in the ATP doesn't actually go into moving the muscle. It goes into the environment as heat. Kind of like in your car, a lot of the energy in the gasoline does not move your car. It's making your engine super hot, which is why you need your engine coolant and your radiator and everything, right? So it's the same thing with your muscles. Your muscles are burning fuel, the ATP, and they're generating a lot of waste heat, which goes off into the environment. However, sometimes we actually take advantage of that, and actually we use that heat for temperature regulation. You know, where do we see that? What's the evidence that we use muscles to actually generate heat to keep our temperature? Shivering, right?
When we
Shivering.
shiver.
You're moving your muscles around. It's not about going anywhere. It's just about making the muscles go, you know, kind of run in place, but then they're generating heat in that process, and you're actually using that heat to try to stay warm. So shivering is actually, instead of having movement with a waste, you know, having some, you know, heat as waste, we're actually using the heat for what we want, and then the movement is just kind of whatever. So, you know, I'm sure you've noticed if you've been running around, whoa, I am now no longer cold here. Or as you keep, if you get on, you start running around, you start taking layers off as you start warming up. That's because those muscles are generating a lot of heat in that inefficient process of transforming ATP into movement and force. All right. So I'm going to start looking at muscles in kind of more of a stand back and look at them. And then we're going to get down into the details of what's going on in the cell to generate force. So again, muscle tissue, I talked about, only contracts. You know, so we should talk a bit about the idea of how muscles are connected to the skeleton. Let's say this is your arm. You know, your muscles, if you did not have your skeleton to work with your muscles, you would only be able to kind of ooze around like an earthworm or something. One of the sad things about the format of this class is we actually aren't going to be talking about bones much at all. And the skeletal system is totally interesting and dynamic. It's not just a armature in your body. It's the key place where your blood is being made. It's living, dynamic living tissue that changes in response to the forces put on it. You know, you break a bone it heals, right? It's alive. So unfortunately, we are not going to be spending time on that because of the kind of format of the way these physiology classes are structured. When I used to teach back at Laney, we had like a 2-semester A&P class where we had more time to go into detail because we didn't have to here, like anatomy, it's mainly anatomy, but you have to talk a bunch about the physiology. And physiology, it's mainly physiology, but you have to talk a bunch about the anatomy. When you do like a joint A&P class, you get to go much deeper into things. But let's just say bones, dynamic living tissue, don't just write them off. They are actually really interesting. And in the context of muscles, though, these are how we are going to have a lot more variety in the kind of movement that's possible in our body. We can take a muscle like your biceps brachii muscle, and we can pull on this. If this is your elbow joint, you pull there and you flex your elbow. That same muscle is not able to extend your elbow. So you have triceps brachii. So if you want to, so it's still a contractile force. The muscle can only pull, but depending on how you attach it to the skeleton, you can actually make the skeleton push or pull. So that's kind of important to remember. Other things important to remember is this idea of levers. If you look at how the muscle is attached to the skeleton, it's usually attached very close to the fulcrum point, the point where the joint is actually swinging or moving. Like on a teeter-totter, if you think of a teeter-totter, if you push a little bit on one side of a teeter-totter, the other side is going to swing really. Maybe I should draw this a little. Like if I have a teeter-totter where it's in the middle here, you push up here, it goes down here. If I have a teeter-totter where it's like here, if I move this a little, this one's going to move a lot. So you have this kind of system with your joints. The muscle moves a little bit. The muscle has a certain amount it can contract here, pulling this up here, but your hand is going really far and fast. So you're kind of amplifying the range of motion and the speed on the actual like skeleton compared to what the muscle is actually doing, right? So, your book's going to talk about different types of levers and things. Don't worry about that. But you should be aware that the concept of levers is important to amplify the range of motion and the speed that things can move based on how the skeleton is attached, well, how the muscle's attached to the skeleton. Other things while we're talking about muscles attached to the skeleton, most muscles have different jobs. Like, your classic biceps brachii muscle, which you usually think of as like Popeye, flexing your elbow. It's actually crossing the shoulder joint. So it can actually flex the shoulder joint. It's actually yanking on your radius bone. So it actually is an important muscle for supinating your hand, for spinning your wrist thumb ways out. So it's actually important that many muscles actually contribute to more than one action. So let me put that in here. You know, so like, again, your biceps brachii muscle, which you usually think of as flexing your elbow. It does flex your elbow, but it flexes your shoulder and it supinates your hand. So that's important to realize. The other thing that's important to realize is most actions have contributions from more than one muscle. So for instance, this idea of flexing your elbow. Flexing your elbow is done by the biceps brachii, but it's also done by brachialis muscle, which also crosses this joint. It's also done by brachioradialis muscle, which also crosses this joint. You know, for extending your elbow, it's mainly triceps brachii, but triceps brachii actually has 3 heads, plus there's little anconeus muscle back there as well. So there's usually more than one muscle that is helping with any particular movement. Sometimes one muscle will contribute more or less at a certain part of the range of that motion. But it also means that there's backup. So if a muscle is damaged, it doesn't mean that you're never going to have that movement again. It means that maybe it'll be not as strong, but other muscles can still help your body move in that particular way. Other thing I will say, here I'm saying muscle only contracts. What I should say more accurately, it generates a contractile force. So let me so it's pulling. It doesn't necessarily get shorter. So what do I mean by that? I can, wow. Well, I thought I had it. Okay, let me get this up real quick. Here, if we this dude, this is like the classic Far Side. This dude is pushing really hard. His muscles are generating a lot of force, but nothing is moving. There's no motion. So it's possible to activate a muscle and generate force, but not actually move, not actually do any changing the length of the muscle. So what do we call this kind of contraction? When the muscle is contracting, generating force, but there's no change in length of the muscles.
Isometric?
Yeah, this is an isometric contraction. So muscle does not change length. Right, iso means same, metric means length. So isometric means the muscle doesn't change length, but it is generating force. The other kind of contraction is isotonic. So it means kind of same force. So we're generating a force, but this muscle does change length. But which way the length changes depends on what kind of force is pulling against it, right? So I could have a muscle pulling in a, let me put my muscle here. Let's put my kind of elbow again. So here's my muscle. My muscle is generating some contractile force. Assuming that my hand is not pulling harder, then my hand is going to go up. The muscle will get shorter. That's called a concentric contraction. But it's possible, what if I'm holding something really heavy and there's a lot of force due to gravity where this is pulling down, I could have this muscle contracting, but it's actually getting longer, right? This is happening if you're slowly lowering something. You're not just letting it drop, but it is still lowering. It's still going down. So it's possible that the muscle contracts, but actually lengthens. So that's called an eccentric contraction. What the heck is that? Let's see. So that's important to realize that you can actually contract the muscle, but it will actually, if you have enough force pushing the other way, the muscle gets longer, but you're still generating contractile force. And again, an example of this is if you're holding something heavy and slowly lowering it down. So you're resisting gravity, but gravity's winning little by little. These kind of contractions tend to cause little micro tears and things in the muscles that stimulate muscle hypertrophy, muscle growing more. So when muscles are experiencing this, it's basically turning on these little messages that tell the body, the muscle isn't quite strong enough to do what we're doing here. We need to beef up the muscle. We'll talk about what that means, like adding more myofibrils and stuff. But if you are doing exercises, like if you're holding a weight and slowly lowering it down, two things will happen. One is your muscles are going to be way sorer than if you were doing exercises where you were winning, because there is kind of more of this kind of micro tears and stuff in there. So this is going to make you more sore and will also, cause the muscles to beef up more. So these are more likely to kind of, if you're trying to bulk out your muscles, you want to do more exercises where you're kind of fighting and losing rather than winning, so to speak, against gravity there. So does that make sense, that the idea of concentric versus eccentric isotonic contractions? Both are changing the length, but you can actually contract and actually have the muscle lengthen. Okie dokie. So now what we're going to do is we are going to get small and talk about individual muscle cells. We're going to talk about kind of the basics of how they contract. We're going to talk about, like I said, different varieties of muscle cells. They are ones that are optimized for like explosive, fast movement, the fast twitch, white fibers, the endurance, red fibers. We're also going to talk about then how the muscle cells are put together in the overall muscle in ways that you can adjust overall force for the whole muscle. And then we'll spend a little bit of time also kind of talking about smooth muscles. So you have kind of that to compare and contrast. Oh, and again, like I said, also we'll spend time looking at how muscles generate ATP. And they have a few different ways to do it depending on how fast they need the ATP. All right. So muscle cells. Sometimes you call them muscle fibers because they're long, thin things. Literally, and again, this is skeletal muscle, so it's got little striations on it. You can have a single one of these muscle cells that can be over a foot long. Like in your biceps, there's single cells that go along the entire length of the muscle. In your like sartorius, the muscle that goes from your hip all the way down to the top of your lower leg, those are single fibers that are going all the way across your thigh. So these can be really long. Embryologically, they start out as lots of cells that all fuse into these big mega cells. They have lots of hundreds and hundreds of nuclei in them. And they're, again, super long. They can be. They can be like, again, over a foot long. So just kind of putting it out there, they're very kind of atypical cells, which is going to be very different. The smooth muscle and the cardiac muscle are more kind of normal mononucleate cells. So this is, again, skeletal muscle cell. All right, so I'm going to start by kind of giving you a basic layout of this, and then I'll shift over into kind of the PowerPoint graphics to kind of speed things up. So I'm going to like do this, meaning like it goes on in both directions. The cell membrane has a special name. Most of the things that have to do with muscles either have the prefix sarco. Sarco means flesh, or myo for muscle. So the cell membrane is called the sarcolemma. So this is and the thing to know about the sarcolemma is that it is full of the same voltage gated channels that we find in the axon. And again, sarcolemma is just the cell membrane of the muscle cell, and it's full of these voltage gated channels. What does that mean? So particularly when we have our motor, let's say this is a somatic motor neuron here. And we're releasing acetylcholine, and it's binding to the nicotinic receptors on the membrane. What is going to happen when we open up the little sodium channels here?
It's going to depolarize.
It's going to depolarize here, and then what's going to happen?
So contracts.
Well, no, we haven't got to any contraction. We're just on the membrane right now.
The action potential is going to go along the membrane.
Exactly. We're going to have this chain reaction, which is just going to go everywhere. Right? As soon as we depolarize a little part of this thing, the whole thing is going to spread like wildfire. Right? That was the whole idea with the action potential. It was this chain reaction. As soon as this little part is depolarized, that's going to open up more sodium channels, so the next part gets depolarized, which opens up more sodium channels, so the next part. So that's going to be important when we look at the whole excitation contraction coupling in the muscle. it is going to rely on the fact that the sarcolemma carries action potentials. Right, an action potential. If we excite one part of the sarcolemma, it's going to spread out across the entire cell. So you keep that in mind. Okay, so there we've got kind of the envelope set up here. It's got this ability, once you excite it at the neuromuscular junction here, that electrical excitation is going everywhere. Huge action potential spreads across. You can measure it. We call it the electromyogram. If you put electrodes on your skin and move around, you can see the voltages flying all over the place. Obviously, we're going to need the actual contractile proteins that are actually going to generate the force. For right now, we're going to just keep them more macroscopic, the little myofibrils. So the myofibrils, which we're going to look at in more detail, you know, the famous actin and myosin, as well as other ones we're going to talk about, the tropomyosin and troponin and titin and stuff like that. But the myofibrils are the things that actually are going to generate the force, the interactions of the contractile proteins within the myofibrils. Other things we need to make this picture or be able to discuss contraction excitation coupling. There's a specialized version of the endoplasmic reticulum within the cell, which we call the sarcoplasmic reticulum. So let's put that in here. So this thing that I've drawn in blue here, this is what we're going to call the sarcoplasmic reticulum. And its main job is a calcium reservoir. It stores calcium. So it stores calcium. So when the muscle is just at rest, there are little ATP powered pumps that, I need a little smaller font there. We basically have little ATP powered pumps that are constantly pumping calcium out of the cytoplasm into the sarcoplasmic reticulum. So this is going to be important. When the cell is, and these are burning ATP actually. So we are actively vacuuming up all of the calcium that is in the cytoplasm and storing it inside the sarcoplasmic reticulum. The calcium is going to be important as well when we talk about excitation contraction coupling. There is a connection between the sarcolemma and the sarcoplasmic reticulum. The sarcolemma has these little invaginations that come in. They're called the T-tubules or transverse tubules. And these transverse tubules are basically where the membrane comes in and kind of plunges in. It would be kind of like if you had a balloon and you stuck your finger on the side of the balloon and the rubber just kind of, so your finger's not really in the balloon. This is still continuous with the outside, but it's like this little bit of membrane that's tunneling down. And again, we're not going to get into the nitty-gritty details, but there are these special little proteins that are connecting between these two membranes here. And the basic idea is when this action potential happens on the sarcolemma, it triggers the release of the stored calcium from the sarcoplasmic reticulum. So I'll write that out explicitly here. So this is going to be an important part of the excitation contraction coupling. Basically, it's, again, I'm kind of putting it together for you where you're going to see, oh, a signal comes down the somatic motor neuron, which then causes a depolarization at the neuromuscular junction, which then creates an action potential across the whole sarcolemma, which then causes a release of calcium from the sarcoplasmic reticulum. And then we're going to see that once we get into the myofibril, that calcium is what's going to allow the actin and the myosin to pull against each other and cause a contraction. So we're going to keep putting this together, but this is at the, these are the first steps. Maria?
One question, Professor. The calcium is released into the sarcoplasmic reticulum.
No, it's stored in the sarcoplasmic reticulum.
Okay,
So
and
it's released out. It's released out of the storage, the reservoir, into the spaces outside where it can interact with the myofibril.
Okay, thank you so much.
Sure. So the last thing we need to do now is look in the myofibril, what's happening to actually generate the actual force. So in the myofibril, most of your muscle cell is just filled with myofibrils, the organelles with the actin and myosin that generate the force. And actually, while we're here, I should mention, when your muscles need to get stronger, you don't get more muscle cells in the overall muscle. You instead, the individual muscle cells fatten up by adding in more and more of these myofibrils. So we have more and more of these organelles full of contractile proteins to all work together. So like if you've been working out and your muscles are bigger than they used to be, it's not because there's more muscle cells in the muscle. It's because the individual muscle cells are actually wider in diameter due to being filled with more actin and myosin in these myofibrils. We'll see also later when we look at muscle cells that are optimized for really strong contractions are just much larger diameter because they have lots more of these myofibrils in them. When you have muscle cells like the red fibers that are optimized more for like endurance and posture and stuff, those are going to be much smaller diameter because they don't have as many of these myofibrils in them. All right, inside the myofibril, we have to introduce some basic proteins that make them up. The thin filaments, actin. Actin is made up of these little subunits which connect up. Again, I'm going to draw it mainly as just a line, the thin filament, but it is actually important to note that there's a little groove in between where all the little subunits connect, and that groove is going to have a binding site for actin. So thin filament actually has little binding sites, not for actin, for myosin, which we're going to meet in 2 seconds. Now there's going to be myosin, the thick filament. Myosin is made up of a whole bunch of these little subunits. Each myosin molecule has a tail, has a head. And actually it's got quaternary structure. What does that mean? It's like has fourth level protein structure. What would that mean?
Multiple protein subunits are combined.
Yeah, exactly. So it has these heavy chains and light chains that connect together to make the complete protein. It's actually got subunits, but then you got this more complicated dealie. And it has 2 conformations. We've been talking about system transretinal has these 2 conformations, 2 different shapes it can be in. Myosin has 2 different shapes it can be in. This is what I would call like the cocked conformation. Imagine a piston. In fact, I can do it. I have these old. Here. This is actually an old revolver from the 1800s. And you have the little thing on the back that you have to pull back, the hammer. This is cocked. And then there's a trigger, and you pull the trigger and the hammer snaps in, right? So here there's a lot of mechanical potential energy. I've pulled the hammer back and it's waiting to go, but it needs the right trigger. You pull the trigger and boom, it snaps in, right? So a myosin head here takes ATP. You need ATP to cock it. It uses energy. And then if you give it the right trigger, the head snaps in. So this is what I'll call kind of more like sprung. This is when it goes boom, it goes in. The trigger for this is the head binding to actin. Triggered by binding to actin. So the basic idea here is you're going to cock the little myosin heads using ATP. They're going to go up, grab onto the actin. As soon as they grab the actin, they'll spring and they'll yank and pull. That's going to generate the contractile force. The myosins are connected together tail to tail to make these, kind of like Q-tips, right? The heads are all kind of outward. So the actual thick filament is hundreds of these little individual myosins all connected together. And then we put these together with a couple of other proteins. So like I said, this is going to be triggered by binding to actin. So if we don't want the muscle to contract, we need to make sure we block the binding sites on the actin. So actin actually has a protein that's associated that blocks the binding sites called tropomyosin. So when the muscle is not contracting, we're going to have this other protein in the way. Let me just draw this out better. What I'm going to draw here, we call the sarcomere. It's basically the kind of repeating unit of muscles. Let me make sure I use the same color coding. I was using blue for myosin, no, blue for actin, red for myosin. Do this more. So sarcomere is basically this, and you basically have it repeating again and again. And I'll show you some better pictures of this. But basically you have the myosin sitting in a little pocket in the actin there. These kind of boundaries where the sarcomeres are anchored to each other are called Z lines. So I should mention your book will talk about all these different terms, A bands and I bands and H bands and everything. Don't worry about any of those. Those were kind of historical before people knew what things actually made up the sarcomeres. They just named them after their optical properties, like isotropic band, anisotropic band. Helle means bright in German. Z is zwischen in German, means between. That is useful to know. So the Z lines are kind of like the anchor points where each of the sarcomeres is anchored to the next band of sarcomeres. We have that tropomyosin in the way here to keep the actin from grabbing on if we are not actually in the process of contracting. For completeness, we'll add in titin. There's actually a protein, the giant protein, this big elastic protein that makes sure that you don't totally pull the myosin out of the pocket in the actin here. And then obviously, what's the other thing we're going to need here? If we actually want the muscle to contract, what do we have to do?
Move the tropomyosin so that the binding sites are available.
Exactly. If we want the muscle to contract, we have to move the tropomyosin so the myosin can grab onto the actin. So that's going to be the job of yet another protein that you're responsible for called troponin. Whoops. Troponin has 3 binding sites. It binds onto the tropomyosin, onto the actin, and it also has a binding site for calcium. So basically, the idea here is when troponin binds calcium, it moves tropomyosin out of the way. So myosin can bind the actin. So this is going to be how contraction works, right? We talked about, if we go all the way back to here, we have action potential on a somatic motor neuron. Comes in, releases acetylcholine, binds the nicotinic receptors, causing an action potential across the whole sarcolemma. That is going to then cause the sarcoplasmic reticulum to release calcium into the cell. Oh my gosh. As the calcium comes into the cell, it's going to bind the troponin, which moves the tropomyosin out of the way. And that means the actin can grab on, the myosin can grab onto the actin. Right? So the basic thing that you have here now, we have the actin, the myosin binds. Once it binds, it's going to pull. That's the thing that makes it go. It's like, boof, it pulls. Right, so that's generating the force. If you go back again to here, all those little myosin heads grabbing and pulling in on the actin is going to basically pull the Z lines in closer together. So this whole thing, they call it sliding filament theory, because it's about filaments sliding. Like the Z lines get closer and farther away, but nothing is really, it's not like a rubber band. It's actually the myosin fiber, the myosin filaments and the actin filaments don't change length. They just get deeper in the pocket or further out of the pocket. So that's important. Right, so when the muscle is shorter, it's deeper in the pocket. When the muscle is longer, they're kind of more shallow in the pocket, but you're not actually changing the length of the fibers. You're just sliding them deeper into the pockets or further out of the pockets. That tightened the, for completeness, I should say that this titin protein right here, we call this titin. This actually does have elasticity. So muscle actually has a bit of elasticity due to this protein that's kind of keeping you from pulling the myosin all the way out. But in general, it's not about elasticity. It's about fibers sliding back and forth. It was a dude named Huxley that first figured this out, the sliding filament theory.
What is the name, doctor? I cannot see it in the here. Thank you.
Do the Z lines get closer to each other?
What's that?
Do the Z lines get closer to each other?
Exactly. The Z lines get closer to each other.
All right.
How do we make sure that when the myosin, okay, we have to go back to this one here. So after this, the next step is you've got to let go and recock. So that's going to be the job of ATP. So this is important though. If you don't have ATP, so ATP is doing two things here. ATP is essential to have the myosin head release. If you don't have ATP, for instance, let's say you're dead. After a while, all the ATP is gone from your muscles. And what happens?
Rigor mortis.
Rigor mortis. You're stiff as a board because all the myosin is stuck onto the actin and the muscles cannot change length anymore, which means the joints can't move one way or the other. So, about 12 to, it lasts for a while until, the muscle tissue starts degenerating. But if you've ever dealt with, somebody who's died, especially if you're gonna set them up for something, you got to move them, in the first few hours. If you wait too long, you're not going to be able to change their position anymore because they're going to be, their muscles are like really super stiff. So rigor mortis is due to this. And again, that recocking, getting it ready to go again. And at that point, it's going to depend on whether or not there's still calcium around. If there's still calcium around, this can reach up and grab and pull again. If there's no calcium around, or if the calcium is gone, then it's just going to be back in a holding pattern, waiting. So let me see. Let me just, again, this idea of excitation, contraction, coupling. This is kind of the fancy word for what I have been, fancy term for what I've been kind of talking about since we started this whole idea. Again, starting with the idea of an action potential coming down a somatic motor neuron. Right, the first thing. Then what happens next?
Releases acetylcholine at the neuromuscular junction.
Exactly. So releases junction, which is gonna, bind the nicotinic cholinergic receptors. Let me spell correctly. On the sarcolemma. You don't need to know the term. The official term, they call it the motor end plate, but you don't need to know that. So what is going to happen now that we have acetylcholine binding those nicotinic cholinergic receptors on the sarcolemma?
The action potential spreads throughout the sarcolemma.
Yeah, so this is going to depolarize the sarcolemma. And AP spreads across the entire stay. Don't autocorrect. So that action potential is going to spread across the entire muscle cell. What is that going to cause?
Calcium release next.
So this causes calcium release from the sarcoplasmic reticulum. And then what happens next? What does that calcium do once it's free in the cell?
Binds to troponin.
Yeah, so that calcium is going to bind troponin, which moves tropomyosin, which exposes binding sites on actin for the myosin heads. And what's going to happen next now that the myosin is raring to go? What happens next?
The myosin heads bind to the actin, the binding sites on the actin.
Yeah, so myosin heads attach to actin. The official form, we say they form a cross bridge. And they are going to snap. So then we get what we call the power stroke. The power stroke is when they snap and pull. So this is going to pull the Z lines, pulls the Z lines in towards each other. So that's the actual contractile force right now because the myosin heads are pulling. And then what happens next?
ATP comes in to uncock it.
Yeah, ATP comes, so release myosin from the actin and recock the heads. Recook. So, and then again, as long as there's still calcium, the system can just go again. As long as there's still ATP and calcium around, they'll reach up and grab and keep pulling. If we run out of ATP, obviously it can't keep going, or if the calcium is no longer around, it's not going to keep going. So what I'm going to do now is show you a video. It's like 4 minutes of your life, but I think it's going to be nice because you can see all of this in much prettier animations that I think will make it more real for you. So we are now going to go here, and here in the module for this week, there is now a fixed link for YouTube link. So here's a link to the animation of the skeletal muscle that, well, I'm going to show you after the next, in 4 minutes from now, this will be true that I showed you in class. Right now it's kind of a lie. Here we go.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
So I think it's pretty nice. Hopefully that helps make it a little clearer as well. After we talk about it, write it out, you can see it visually.
Yeah, I definitely did. That helped.
So one thing that that showed that we should probably elaborate on, that kind of had the actin, and it showed the myosin grabbing and pulling and then letting go and grabbing and pulling. In reality, if it was just like that, we'd have trouble. Like if this is like my sarcomere, and here's my myosin in there. If these things grabbed and then let go, and then grabbed and let go, and grabbed and pulled and let go, imagine if you're trying to do tug of war. If anybody, I don't know if people still do that, tug of war, where you get a rope and everybody's on either side of the rope and they're all pulling and trying to see who can pull that other side over to their side. If everybody let go of the rope at the same time to kind of reposition their hands, the whole rope would just slide out from their hands, right? So what has to happen during muscle contraction to make sure that the muscle doesn't just pull apart when the myosins are letting go to regrip?
Are they others attached, like it doesn't happen simultaneously, just alternate?
Exactly. The same way if you're playing tug of war, not everybody lets go of their hands at the same time, right? It's like one person repositions, but everybody else is still holding. So in sliding filament theory, you know, one of them will let go and then re-grab, but the other ones, so it's not happening simultaneously, or else you would have the trouble of the muscle sliding out on itself. So that's the basic idea of sliding filament theory of muscles. So things we're gonna now shift is different ways to actually generate the ATP. Obviously, you need ATP to have this thing going. And depending on how many myofibrils, you might need more or less, right? If I have a lot of myosin to detach and recock, which is going to happen in muscle fibers that are really fat with lots of myofibrils optimized for a lot of strength, I'm going to need a lot of ATP stat to keep this thing going. If I have a muscle that's a little more leisurely, it's like, let's keep pulling, but it's not like tons of myofibrils that all need ATP right now. Then I can be a little more leisurely in my generation of ATP. So let's take a look at the different ways that muscle fibers generate ATP, and then we'll talk more specifically about different varieties of muscle fibers that will feature one or another of these methods. So, kind of making the ATP in the muscle cell. What's the kind of baseline way that cells make ATP?
Respiration.
Cellular respiration.
Yeah, exactly. So cellular respiration is kind of the gold standard for making ATP. It's going to be the most efficient in terms of converting the energy stored in the glucose into actual ATP molecules. It's got a couple of drawbacks though. I mean, the main drawback is it takes a billion steps, right? You saw that. It's like, go through the 10 steps of glycolysis and then move into the mitochondria and then go through the Krebs cycle and then pump the protons and let the protons come back down. And there's a lot going on before you finally got the ATP in your hand. So in these muscles that are optimized for really fast, strong contractions, they want to come up with ways to make ATP faster. So one thing you can do is just do glycolysis. So if you remember glycolysis, how many net ATP did you make after you were done with glycolysis?
Two.
You only make 2, but you make them really fast. It's only 10 directly connected steps. Zing, you've got 2 ATP. So it turns out if you are just interested in the most ATP per second, and you don't care that you're actually wasting most of the energy stored in the glucose plus making lots of lactic acid as a byproduct, this is actually way faster. This is about 2.5 times faster. If we're looking at ATP per second, how many ATP per second are you getting in the cell that you can actually use? If you just stop at glycolysis and just use the ATP directly right there and forget about the rest, it's way faster than cellular respiration. You know, the downside is you're going to build up lactic acid. And also it's only going to have the sugar that is available in the moment. we're going to see this, the muscle cells that are optimized for using glycolysis are going to have little glycogen granules just stored within the muscle cell itself. So you have kind of a supply of sugar ready to go. And it's only going to go for as long as you have sugar in the muscle. This lasts like maybe, you have like maybe 1.5 minutes of this before you run out. And also you're building up the lactic acid, which has to get cleared out. The lactic acid gets like transferred to the liver, where it gets kind of, I think, turned back into pyruvate. So, and there's yet another one. If you need sugar even faster, not sugar, ATP even faster, there's what's called the phosphagen pathway. This is going to make ATP 4 times faster than waiting around for cellular respiration. And the basic idea here is you have this molecule called creatine phosphate. Which I'm just going to draw as like a C with a little phosphate on it. So you've got creatine phosphate sitting in the muscle. And in the muscle, we have ATP getting broken down into ADP plus phosphate, right? So we're building up this ADP as a byproduct of Actin. That's what I meant. ADP as a byproduct of breaking down ATP, and it turns out we can regenerate it in one step by using our pal creatine phosphate. So basically the idea here is creatine phosphate plus ADP all we need to do is transfer that phosphate over to there in one step, and that is going to give us ATP. You know, and then the kind of creatine, which now has to get regenerated. So in one step, we have a new ATP. What kind of enzyme do we use to transfer phosphate groups like this?
Kinase?
Kinase. So we have a creatine kinase that does this. So we have this enzyme which will facilitate the transfer of a phosphate group from this creatine phosphate molecule. And in one step, we got new ATPs. This only lasts, we have about 10 seconds or so worth of creatine phosphate ready to go. So if you are like just doing like a 50-yard dash and really sprinting hard as you can, you're probably purely in this mode. If you kind of keep going, you'll start getting into here. And after a while, you've used all of these up. And then you have to regenerate them. Both this and this need to get regenerated after you use them. You need to replace the creatine phosphates because you've just used them all up. You got to get rid of the lactic acid, and that's been building up as part of this, and regenerate also the glycogen and stuff in the cell. So this and this will last about 10 seconds. This will take you up to about 1.5 minutes, but then they're done. And it takes actually a bunch of extra oxygen, because neither of these are both anaerobic, right? You don't need oxygen to do glycolysis. You most definitely don't need oxygen to just swap a phosphate group from one molecule to another here. But you are incurring what they call the oxygen debt. The idea of the oxygen debt is once you have used the capacity of glycolysis and the phosphagen system to make lots of ATP fast, it's gonna take a bunch of extra oxygen on top of your normal oxygen to do the processes to recover from them. So here's the idea of oxygen debt. You need around 11 liters extra oxygen to kind of, recover from using those anaerobic pathways. So you've probably noticed this, right? You're running and running as fast as you can, and you've got the energy and you're doing it, and then you stop and you sit down and you're going, like you're breathing, but you're not doing anything. You're standing there. You're not, muscles aren't, they don't need ATPs right now. You're just standing there hunched over, breathing heavy, right? That oxygen is not being used to run anything that is happening at the moment in terms of your muscles. Instead, that oxygen that you are desperately trying to suck down is this oxygen debt. It is the stuff that is recovering and regenerating those systems that you used in your muscles during those anaerobic pathways. So that's what that oxygen debt is. That's also why you're breathing heavy when after you've pushed your muscles hard, even though you actually aren't moving your muscles anymore. You've incurred this oxygen debt. So does this make sense? If you've got the time, you'd rather just do cellular respiration. There's no oxygen debt. It goes on. You don't have to store anything necessarily because the blood keeps bringing in sugar and oxygen. So, but again, if you need a lot of ATP fast, this doesn't have the capacity that you need. So then we have these other systems to make ATP faster so you can run those myosins quicker and have more kind of a higher capacity of muscle contractional force. We're going to, again, talk about like the slow twitch fibers. The red fibers are going to be optimized for the cellular respiration because they don't need a lot of force. They're more optimized for endurance and not force. Whereas the muscles that are optimized for force and speed are going to be have specializations that make them especially good at these anaerobic pathways. The white fast twitch fibers are going to have lots of creatine phosphate and glycogen granules to do these anaerobic pathways. So that's making the ATP. So in fact, this is as good a time as any to introduce the idea of fast twitch and slow twitch muscle fibers. So types of skeletal muscle cells. So all of the skeletal muscle cells do the basic stuff we've been talking about, the actin and myosin, sliding filament theory and all that. But there are different versions of them. Kind of like there's a bunch of different kinds of dogs, but there are, dachshunds that are optimized to go down holes and hunt badgers. And there are, other like Doberman pincers or, German shepherds designed to guard things and bite you if you're trying to be where you're not supposed to be, or whatever. There's different dogs that are optimized for different kinds of jobs. So there are different kinds of muscle fibers that are optimized for different kinds of jobs. And there's a variety, but we'll focus on kind of the two main divisions we talk about. red fibers versus white fibers. White is also known as fast twitch. White is also known as glycolytic. These are kind of all synonyms. Red is also called slow twitch. It's also called oxidative. And you'll see why these names make sense. The red, their color, they have red color. Slow twitch, they're going to contract more slowly. Oxidative, they're going to use oxygen. The white, fast twitch, they're going to contract more quickly. Glycolytic, meaning they use like glycolysis, these anaerobic pathways. So let's look at each of these in more detail. So one is the color, although we'll get to the color a little later. So let's talk about the slow versus fast. These contract more slowly. And with less force. These contract more quickly, with more force. Part of this is due the speed of the contraction actually has to do with slightly different versions of myosin. The myosin in white fibers actually snap quicker. So they are going to actually be pulling faster than in the power stroke than they do in red. The force is more about how many myofibrils you've got in the cell. These red fibers, these are much smaller diameter. Smaller diameter, due to less myofibrils. These are going to have a larger diameter, right? If you have more myofibrils all pulling in parallel, you're going to generate more force. Other things that are different about these, the red fibers have, because you might say like, whoa, what a bummer thing, red, like slow and weak. Why the heck would I ever have a red fiber when I could have the quick, strong fibers? It has to do with endurance. These are, good for endurance. These fatigue quickly. Red fibers keep going and going and going. These are the ones, like if you have muscles that are part of your like posture or something, whether you just want to keep holding things, you don't need a lot of strong or really fast, like, contractions, but you want to just keep going and not get tired. That's what these red fibers are optimized for. These white fibers fatigue quickly. They can generate a lot of force quickly, but they're using those anaerobic pathways that we've talked about. So, within a minute or so, they're pretty much spent and they need to recover. One of the ways to think about this in terms of things you're familiar with is think about a chicken. What's the chicken's like normal day? Normal day is, pecking around the yard and, oh my God, there's some seeds or there's a worm. So it spends a lot of time walking around. So its endurance muscles are going to be in its legs, where it just kind of keeps going. What kind of meat do you find in the pulkeys, in the drumsticks of a chicken?
Dark meat.
Fibers.
That's the dark meat, which is basically the red fibers, because its legs are built for endurance. If you go to the breast meat, which is about the wings, what kind of fibers do you find there?
White fiber.
That's the white meat. Like how does a chicken use its wings? Does a chicken gracefully fly through the air? It's like, whoa, there's a flock of chickens up above the lake.
It needs energy quick and fast.
It's quick, yeah. I mean, if anybody's ever seen like Rocky, that old movie, Rocky, part of his training, he has to try to catch a chicken, right? And he goes and he tries to grab the chicken, and the chicken like explodes and flies away, and then kind of lands someplace a little far away, and then kind of keeps pecking, right? A chicken's wings are going so they can like get away, fast, explosive, but it's not about sustained endurance. What about a duck though? Has anybody ever eaten a duck? What does the breast meat look like in a duck?
They're very fatty.
It's red.
It's red, it's dark. You have dark meat in the breast meat of a duck, right? Because a duck actually does need endurance for flying. So it's gonna have this endurance muscle that can just keep going and going and going and going, and they can just stay up in the sky, as opposed to chickens that are optimized for that, basically a sprint. Their wings are like sprinting, but not for kind of long distance. So just so that's the basic idea of like red and white, dark meat and white meat. Other things, because these have less myofibrils and aren't going to be as ATP hungry, these tend to use cellular respiration. To make ATP, and because it's good enough, which means they're going to have, more blood vessels to bring the oxygen in. They also have a pigment. The reason they're red is they have this stuff called myoglobin, which helps bind oxygen in the tissue. So myoglobin is like hemoglobin, except it's found in the muscle, which is why it's called myoglobin. So myoglobin binds oxygen. That's why it's reddish. But so good for endurance. Uses cellular respiration to make the ATP, which means it's going to have more blood vessels, and it's got this myoglobin, this reddish stuff in there to bind oxygen. The white muscles, these are optimized for anaerobic respiration. They're going to need, because they are larger diameter, more myofibrils. They're going to need a lot more ATP right now, which means they're going to use the anaerobic pathways. These are going to have creatine phosphate. So they can do that phosphagen system. They're going to have glycogen granules. Right? And what is glycogen? Everybody should remember what is glycogen?
Store glucose.
Basically store glucose. It's like animal starch. It's like one step you're freeing up glucose for the cell to be able to use. So glycogen, like when you're carbo loading before like some race or something, what you're doing is like trying to get lots of glycogen like stored in your muscles and stuff. So it's waiting for you. So these are the two main types of muscle fibers. Some muscles are predominantly one or the other. Like I said, like muscles that are used more for posture tend to be of the red type. Things that are more about just explosive sprinting or more of this. A lot of muscles are a mix, and depending on how you train, one or the other one is going to be more fully developed, right? If you are doing a lot of kind of heavy weights with low reps, you're going to be working on building up your white muscle fibers. If you're doing like high reps at low weight, endurance kind of training, you're going to be building up more the capacity of your red fibers. I should also mention the distribution of red and white fibers in a particular muscle is genetic. So no matter how you train, different people are going to have a different level of ultimate performance. For somebody, the same training, they're just going to be better at sprinting, or another person's going to be better at distance running, because the distribution you have of red and white fibers to start with is genetic in your muscles. What else? I should also mention there is a continuum. There are some cells that are like, there's fast oxidative fibers. There are fibers that tend to be like faster contraction and yet still more efficient and don't fatigue as quick. So there is a continuum. But you know, for right now, these are the two main types of fibers: the red fibers, red slow twitch, white fast twitch. And again, fast twitch because they contract quickly with a lot of force. Slow twitch because they contract slowly, less force. So any questions about those? So now what I can do actually is bring in twitch. So I'm saying fast twitch and slow twitch, but what? So twitch is not just like a goofy word I'm using here. It's actually, there's a formal definition of what we mean by a twitch. So let's look at what a twitch is, and then talk about how it leads to an interesting problem for how we generate force in a muscle as an overall organ. Okay. So in this graph, this is an unspecified unit of force. It's how hard basically the muscle fiber is pulling. So when an action potential comes down a somatic motor neuron, there's going to be a little bit of a delay, right? So first thing is depolarize the sarcolemma, release the calcium, calcium binds the troponin, the tropomyosin moves out of the way, the myosin grabs on, and power stroke. So you get some force. There is going to be temporal summation. If I am but when you, the way you typically, so this is called a twitch. And it's an all or none kind of thing. You can't make it more or less. It's just kind of a, based on the kind of like how many myofibrils are in the cell and all that. So you do an action potential and there is this ballistic sequelae of events that happens where things open and bind and pull and all that. Typically what you do, if you just keep firing action potentials down there, it comes in, the calcium builds up more and more of the myosins are all going together. So this, if you keep firing off, you are going to get this maximal sustained contraction for a fiber. and that is called tetanus. So this is not like the disease tetanus where you are paralyzed or something. This is just the formal word for if we fire off, we keep exciting this muscle cell so it's flooded with calcium and all the myosins are working together. This thing is in tetanus. And there is this maximal sustained contraction, and it's going to be defined by the mechanics of that cell, how much actin and myosin is in that cell to be pulling. If you wait after a while, it starts going down. This is called fatigue. You stop generating force. Even though we keep exciting it, it starts losing the ability to generate force. This is going to be important as well. You know, one of the things I've talked about how, you know, we're still learning something as core to our physiology as fatigue. Like, how come the muscles start getting tired if you keep trying to push them? We don't totally understand. In fact, the story has changed since I've been teaching this class. They've come up with a couple of different hypotheses about what they think is happening in fatigue. And then they realized, no, that isn't actually, with more data, that's not supported by the latest data. We're still not exactly sure why muscles fatigue. You know, this whole idea, remember the sliding filament theory. You know, the idea, what is a theory? A theory is, you know, the preponderance of evidence. You have a hypothesis and you support your hypothesis, and enough people kind of get evidence that keeps supporting the hypothesis, you know, then it's starting to become a theory. A theory is something where there's more and more evidence that isn't contradicting itself, saying this really does seem to be, it's no longer like a supported hypothesis. We're kind of elevating it to the next level of it's the theory. But that doesn't mean it's true. It means like this is our current best understanding, and it's subject to revision with more and more information. You know, sliding filament theory, parts of it really don't change at all. Like the idea that actin and myosin are sliding past each other and pulling to generate the force. Like I'd bet my life on that one. That one is pretty well, it'd be really shocked if that wasn't the truth. But when you get to the more subtle things of muscle physiology, even something like how come the muscle starts like losing force if you keep exciting it? We're still trying to figure out some of this more subtle details of how this works. So just kind of putting that out there, like that our understanding of the body is a work in progress. And again, that's part of the reason why I go so deep into this process of science and lab, because as you go off into your clinical, careers, you're going to constantly realize that recommendations are changing, or understandings of what exactly is going on here is changing. And the more you understand that process, the more you'll be able to kind of surf that with kind of grace. So this fatigue, and I should also be clear that this fatigue is a physiological loss of force. This is not like central fatigue. We also talk about fatigue in the central nervous system. Central fatigue is like, I'm tired, man. I just want to just plop on the couch and not move. But that's nothing to do with your muscles not being actually able to generate force. You just don't want to turn them on with your nervous system, right? So central fatigue is another thing. That is a whole other thing of like, I'm feeling tired and I just want to sleep, or I'm tired and I really don't even want to like move to push that button. But that doesn't mean you couldn't if you manage to like convince yourself to do it. So fatigue. All right, so now what we're going to talk about is this. So this is a muscle fiber we've been talking about. And if you start turning on a muscle fiber by sending action potentials down the neuron that activate it, you are going to get this contraction. But that contraction is what it is. You can't make it stronger or weaker. It's based on just the actual like kind of anatomy of that cell. How many myosin heads are pulling on how much actin. So a particular muscle fiber has, when you turn it on, it pulls with a certain strength. But obviously you can, in reality, have lots of different forces that you use your muscles. When the overall organ, which is made out of lots of different muscle fibers, you can pet something, you can whack things, you can do a lot of force, you can do really gentle force and caress or punch a hole in a wall or whatever. So what we need to talk about is when you look at a muscle overall as an organ, how do you generate this broad range of force, even though an individual muscle fiber is basically either on or off? So, and the key to this is going to be motor units. So a motor unit is basically a motor neuron associated muscle fibers that it innervates. So if I am looking at an overall muscle, so this is my overall muscle. Let's say it's your biceps muscle or your triceps muscle or your gastrocnemius muscle or whatever muscle. This is the muscle that is connected to the bone. That's the big organ made out of lots of different muscle fibers connected. So a motor unit is going to be like this. There's some neuron. Let's start with the red motor unit. There's some motor neuron, and it's going to innervate a collection of muscle fibers within here. Let's just do a so here's some muscle fibers, and this red motor neuron is doing those. Let's do a different color one. Let's do a blue motor unit. Oops. So here are all my blue. Whoop, let's do this. This is coming down and it's innervating that one and that one and that one. And then maybe we'll have a green one. So this is illustrating motor units. there are different motor neurons, and each motor neuron is activating a collection of muscle fibers, muscle cells that make up the overall muscle, right? Each of these things are muscle fibers, muscle cells that I'm drawing. And the basic idea is, if you want, even though each muscle cell is either basically on or off in terms of how much force it generates, you can, it's called recruitment. You can recruit more or less motor units depending on how much overall force that you want to have this muscle to have. Right? If I want to have a gentle contraction, I want to caress somebody's face, I'll just turn on a couple of these things and have a very gentle pull. Let's say I'm trying to, again, punch a hole in a wall. I want as much force as I can get. I'm going to turn on all of these, and all of these fibers are going to be working in parallel and generate a very strong force. So by recruiting more or less motor units, you can have a greater or more fine force that you're generating. How many muscle fibers do you think we have in a particular motor unit?
Wouldn't it depend on what muscle it was?
Yeah, it totally depends. Exactly. Right? So if you have a muscle where you need really fine control, say like in your fingers, or the muscles that move your eyeballs around, you want to be able to add just a little more force or just a little bit less force to have really fine control over the amount of force. There might only be 3 or 4 muscle cells per motor unit. And you can add in the force plus or minus a few muscle cells at a time to have this really super fine control over the force. You get to something like the calf muscle, like the gastrocnemius, where you just have to either be on your toes or not, or bounce around. There might be 500 cells in a motor unit, because there you don't need fine control. Typically in a muscle like your biceps or something, it's somewhere around like 50 to 100 muscle cells per motor unit. But like I said, it can be a range depending on how much control you need over that particular muscle. While we're at motor units, you know, I have, here's like the little Choco Cat tape dispenser again. I'm kind of holding it up here for all you to see. And I am holding it, and it needs muscles. I have my biceps, I can actually touch it. It's kind of tensed here. Actually moving my whole body in a position to hold my body so you can see it just right. And I could sit here for a long time. I could actually sit here for the next half an hour and just keep showing you the Choco Cat tape dispenser, even though my muscles need to contract to keep my body position proper. And we know though that muscle fibers, if you just keep contracting the muscle fiber, it goes into that fatigue and starts to lose force. So how can I keep this pose and keep the little tape dispenser in your field of view, even though the muscle fibers are fatiguing and starting to lose force?
Do you start to recruit more of the motor neurons that are attached to like the slow oxidative fibers?
Or you support?
So it's more about what we call asynchronous recruitment. like, let's say I'm using this one right now, and these fibers are starting to fatigue. I can just kind of rotate. Let's let this one take over now and let these ones recover. And then when this is fatiguing, let's let this one take over and let that one recover. So this idea of asynchronous recruitment means that you have different muscle fibers that are kind of rotating in and out. So one group can recover while another one takes over. So we never actually see the fatigue happening. Assuming what happens if I, if my pose is I'm going to lift this chair up and show you the chair, and I'm going to try to hold this for a half an hour. It's like, I can't do it. I have to put it down. How come I can't hold the chair up there for a half an hour?
Because you're using a lot of cells at the same time, so they're all going to fatigue at the same time.
Yeah, exactly. If I have to use a whole bunch of motor units to start to generate the force, then there's not going to be any left waiting in the wings to take over when those fatigue. So I'm going to just be tired and I got to recover. So that's motor units. Motor units are these things that are helping you generate more force, less force. Let's see what else I have about skeletal muscle that I want to. I know, we go back. Red and white. If red fibers are optimized for cellular respiration, in addition to more blood vessels, what else are they going to have more of?
Mitochondria.
Exactly. I forgot to put that in there. These also have more mitochondria. So that's another difference. White fibers are not going to have the mitochondria because they're not optimized for cellular respiration. If I took somebody, we're not going to do this today, but let's say you put a blood pressure cuff on them and you pump it up so much that you block all blood flow. So the blood flow can't make it down to the hand. How long can they keep moving their finger and hand around, do you think, before it'll stop working? Can I go back to
Like 3 minutes, because you can't switch over to cellular respiration after a certain time because you need oxygen.
Yeah, exactly. So you'll be able to work your muscles for a while, for a few minutes without oxygen. But after that, everything's going to stop working. Because once you have used up the anaerobic pathways and the aerobic pathways can't work and you can't regenerate things to kind of recover, then the muscles are going to have to wait until the blood flow resumes to actually function again. So that's, so exactly. You've got a couple of minutes of actual muscle activity before things shut down. What else do I want? So are there any questions about skeletal muscle?
What's the definition of a twitch? Once again,
A twitch is basically that whole excitation contraction coupling thing. It's muscle is excited and there is the release of calcium and generation of force, and then it resolves.
One question, how Can you go over the lactic acid?
So if you are doing this glycolysis to make ATP in your muscle, then let me get a better. If you're doing glycolysis, you're making ATP fast, but as we saw from, our study of kind of, glycolysis in the beginning of the semester, you're also making lactic acid when you're doing this just as an anaerobic pathway. That lactic acid ultimately has to get removed and converted back into, I think it gets converted back into pyruvate. But so this gets picked up and moved back into the liver where it gets converted back into something you can use, because you don't use it directly. So you have to, as it builds up in the muscle, you need to you need to transport it out of the muscle so you can deal with it. Well, Emily, let's stop you there. When I first started teaching this class, people were thinking that the buildup of lactic acid was lowering the pH and that was causing inhibiting the release of calcium, and that was causing fatigue. And then they found out actually low pH doesn't seem to have any problem at all with the release of calcium. So the lactic acid isn't directly stopping the cells from functioning. But there's a lot of detail about muscle physiology that I'm not as familiar with. So most of what I know is that it's getting built up and then has to get taken out and
It gets taken to the liver and reused up there, right?
yeah, and the liver has all sorts of, we're going to talk about the liver later, but the liver is basically like this crazy chemistry set which can interconvert things from one thing into another. So the liver takes stuff and detoxifies things, converts things from one thing into another, takes lactic acid and turns it back into things you can feed into glycolysis. So that's all I'll say about that. Anything else about muscles? I never talked about the word muscle. Where does the word muscle come from? Comes from musculus. Anyone know what musculus is in Latin? It's kind of what moose. It's what it kind of sounds like.
Like male?
What's that?
Sorry, I was thinking musk as in male or.
No, it's moose as in mouse. It means little mouse. Again, I think it's because if you, let's see. There, see where my muscles are moving there? It looks like there's a little mouse running up and down inside under my skin in my arm there. I can make it bigger. Let's see. Boop. Right, so let's see. Yeah, you see, it looks like little mice are running up and down underneath my skin there. So it's kind of the bizarre origin of that word muscle. It means like little mouse. What else do we want to say about muscles? Or those kind of muscles. Also, again, I'll reiterate the idea of electromyogram. Maybe I'll write that word down specifically because you should know it. EMG. Electromyogram. myo is just muscle. Again, as your muscles contract, there's big action potentials going across the sarcolemma, and you can measure those voltages on the skin. And they actually can be used, they're used in research a lot. They can be used clinically as well. It's the same idea. Remember, we had the EEG, the electroencephalogram. It was the electrical activity measured on the skin from brain activity. Next week, we're going to be looking at the electrocardiogram. Cardio, heart, electrical activity measured on the skin. That is the result of electrical activity of your heart muscle. So electroencephalogram, electrocardiogram, electromyogram, those are all measuring voltages measurable on your skin that are being generated by, deeper structures, either the muscles or the brain or the heart. All right, so we are going to tie off our marathon muscle day with a little bit about smooth muscle. So again, this is going to be a lot shorter than our discussion of skeletal muscle. So smooth muscle. All right, it gets its name. Yeah, because my little silly drawing. It's not because it's just like really like kind of suave. It's more about it not having the stripes that you see in striated muscle or skeletal muscle. That doesn't mean it doesn't have actin and myosin. It has actin and myosin. It's just not laid out in that regular pattern that we see in the sarcomeres. So actually, so before we get into the detail of a smooth muscle cell, let's talk about just some of the overall differences between smooth muscle versus skeletal. Right, one is it's involuntary. Not that skeletal muscle can't be involuntary, right? When somebody whacked on your patellar tendon, you kicked out, that was an automatic reflex. Obviously, skeletal muscle can be part of involuntary activities. But as a rule, smooth muscle is the muscle that you are not consciously controlling. Involuntary, it's typically found, the walls of hollow organs. Right, it's making up the walls of your whole digestive tract, your stomach and your intestines and your throat and your esophagus. It's making up the walls of your bladder, of your uterus. It's making up the walls of all your blood vessels. So it's typically the walls of hollow organs, so those organs can then kind of squeeze and relax and do different things. Also other places, also like, the iris, erector pili to give you goosebumps, etc. So there's, that's the wrong way to spell etc. So obviously lots of other places as well, but one of the big places you find it is in the walls of hollow organs, but then other little places where you need to pull or squeeze. And again, this is all under autonomic control.
So the muscles found in the ear, those tensor tympani and the stapedius, those are also.
No, those are actually, I think those are actually skeletal. And like your diaphragm is skeletal, even though in general you don't think about contracting your diaphragm. You don't go breathe, or if I better keep remembering to breathe or else I'm going to die, right? So as a rule, though, most of the muscles that you don't think about tend to be smooth. They contract slower than skeletal muscle, but so like maybe 30 times slower, way slower, but can sustain contractions with much less ATP. Right, so you don't, because if you think about what these muscles are doing, is a lot of times they're just like, in your blood vessels, they're holding the diameter. They just have to lock in. Or the sphincters, like right now, the reason you're not pooping in your chair is you've got your sphincters closed down and keeping all those tubes, shut off. And they just keep squeezing and squeezing and squeezing. You don't want to just be burning lots of ATP to keep holding. So there's like this special way that the myosin actually kind of latches on that lets you sustain a contraction with much less constant flow of ATP compared to skeletal muscle. So that's another big difference. And then the other big difference is you can stimulate it to contract in many different ways compared to skeletal muscle. So one way is by a motor neuron, like skeletal muscle, autonomic motor neuron. We will talk a little more about this in a few moments because the neuromuscular junctions in the, good God, hold on a sec. The neuromuscular junctions in the smooth muscle look a little different than the neuromuscular junctions in the skeletal muscle. But it can be activated lots of different ways. It can be activated by hormones. Like I said, think about epinephrine, adrenaline actually can operate smooth muscles as part of your sympathetic nervous system. Different chemicals. When we get into control of blood flow, we're going to see that just levels of oxygen or carbon dioxide and things like that can actually affect the smooth muscle in the blood vessels. So just chemicals floating around can actually create or relax muscle, can contract or relax these muscles. They kind of co-stimulate each other. Right, these are all connected with gap junctions in a particular region. So if one gets excited, if you remember, gap junctions cause an electrical connection. So if one of them is excited, the whole group gets excited. Most smooth muscle is what we call single unit. Rather than having a motor, all these different motor units like we just talked about in skeletal muscle, it's usually the whole group contracts or it doesn't contract, rather than this motor unit or that motor unit. There are a few exceptions we'll talk about, like in the muscles that control your your eyes, for instance, actually are multi-unit smooth muscle. But for the most part, a lot of it is just single unit where all the cells either all contract together or they don't. It can get stimulated to contract just by stretch. So this is actually going to become important when we, again, look at autoregulation of blood flow when we get to the cardiovascular system. If I have some blood vessel here with smooth muscle in the wall, and I push out from the inside, and stretch these muscle cells, the response is this whole thing is going to push back at me and squeeze in. So this idea, if it would be like if the wall was made out of smooth muscle and you pushed at the wall and kind of stretched it out, and all of a sudden, wah, it pushed back at you. So the idea, if you stretch this stuff, it ultimately opens up calcium channels and stuff, which is going to cause the whole contraction to happen. So these are things that distinguish smooth muscle from skeletal muscle. It can be stimulated to contract many different ways, not just from the nervous system. And like I said, it contracts much slower, but can sustain a contraction with much less ATP. So then I'll just talk a little bit more about the actual kind of structure of the cell itself to give you a little more sense of how is it that actin and myosin are working together, but there's no stripes. So they're single cells as opposed to the skeletal muscle cell where you had these big crazy weird cells with many hundreds of nuclei. A single little cell, and it does have actin and myosin, but it's laid out really different. So it's kind of more in this lattice. The actin is anchored by these little things called dense bodies. And again, you can kind of think of them, they're kind of like the Z lines. In that when the myosin pulls on the actin, all the dense bodies gets pulled towards each other. But as you can see, when this thing contracts, it's not just going to be getting shorter. It's also going to kind of get bulgier because it's kind of pulling in all these different directions. So, but this is why smooth muscle does not have stripes like striated or skeletal muscle, because the actin and the myosin is laid out in this grid rather than just alternating in a linear way. There are other differences we're not going to go into, like the calcium. A lot of the calcium actually comes in from outside the cell during the contraction rather than just being stored inside a sarcoplasmic reticulum. And then the other big difference that I mentioned is all of these cells are connected with gap junctions. Which means if one cell gets excited, all its neighbors get excited as well. Right, so if this dude gets it back, that's not what I meant to do. If this guy gets excited, all of a sudden the gap junctions means these guys get excited. And if these guys get excited, that means these guys get excited. So whatever electrical excitation happens is going to spread through the whole group and they're all going to contract together. The autonomic neuromuscular junction is a little different from what we saw with the skeletal muscle, and it's kind of related to this. So this is an autonomic motor neuron. rather than have this real distinct neuromuscular junction, instead they have these little, they're called varicosities, these little swellings that release the neurotransmitter and basically over the whole group of cells. So rather than one motor neuron talking to a specific cell and having this very direct connection, it's more diffuse. They call these diffuse synapses. Because you're basically puffing the neurotransmitter kind of over the whole group of cells rather than having a specific neuromuscular junction. So does that make sense? So, and I'll just, the thing I said in words, I'll write down in letters. Single unit, meaning there's no motor units. That is just one big group. You either activate it or you don't activate it. There are exceptions. Some is multi-unit. And for example, iris and ciliary muscles. So for instance, the smooth muscle that changes the diameter of your pupil or changes the focus of your lens, those actually are multi-motor units with smooth muscle, but that's not so common. But I just wanted to put that out there for completeness. So any questions about smooth muscle? Okay. Are people interested in me just kind of reviewing a bit? Yes, no, yes, okay. So, and I should say, like in 45 minutes, there's no way I'm going to cover a month worth of material. So if people have specific questions or specific things that they would like me to go over or look at, they always say this in all these meetings I go to, and I should do it. Write it in the chat and I will try to integrate it into the, or you could just raise your hand or say something, but I will try to come, Lauren?
Well, if you're done talking, I have a question.
Sure.
So with all the parts of the brain that we went over, like the epithalamus, hypothalamus, all those things, do we need to know like kind of how they interact with each other or like what they're involved in? I guess kind of how deep should I be reading into all that in the book?
I mean, you should know their basic roles for sure. Like epithalamus, there's not a lot there. It's where you find the pineal gland. You should know the pineal gland makes melatonin, and melatonin's involved with kind of circadian rhythms. Probably not a bad idea to know there's a choroid plexus there, because then that takes you into what's a choroid plexus and how do they connect into everything. The thalamus, we really didn't talk much about it beyond it's a relay station for sensory information. And then we implied it's doing more. I talked about it. Oh yeah, it's actually a little bit involved in the limbic and stuff. Hypothalamus, that one had a million different things it did. So
Okay, that's kind of, yeah, what we went over in lecture. So I just wanted to clarify.
So kind of like at that level, I mean, the level I went over in lecture, there's so much stuff and so many connections.
Yeah.
For the cerebral cortex, you should definitely know the basic primary regions we talked about, and kind of what happens there. You should know some of the deeper gray matter and what they do.
The anterior cingulate gyrus and the insula, are those part of the cortex, or are they deeper nuclei? Or
They're both cortex.
Okay.
They're kind of more hidden. If you remember, like if this is the brain from the side as we kept drawing, if we cut it this way and we look at the two hemispheres, with all the other stuff inside, when you're just looking at the brain from the side, you don't even see them because the cingulate is in that central fissure there, that longitudinal fissure, kind of right above the corpus callosum. The insula, you actually have to go down to the bottom of the lateral fissure here, and the insula is down in here. So they are officially cortex, but if you just pulled the brain out of somebody's skull and held it in your hand, you wouldn't see them necessarily right away.
Okay. And then the like hearing and vision stuff, is there going to be some overlap with the lab information? Like, do we need to memorize like someone with hyperopic vision and?
So there's no hyperopia or ametropic eye.
Okay.
More what I'd say more what you want to focus on. You should know there's the cornea, the lens to do the basic focusing, but then the stuff we really added in was all the actual sensory transduction. Like once it hits the retina, how do we actually go from light hitting a cell to creating an electrical signal? That's really what we focused on for the lab part. You should know it's processed in the occipital lobe and the primary visual cortex there. You should know the details of how we convert light to an electrical signal in the rods and cones. But we're not going to get into the details of either Snell and eye chart or what's astigmatism or anything like that.
Okay.
Hearing, you should, you don't need to know a Rene test or a Weber test, but you should know that you've got a tympanic membrane, vibrating ossicles, vibrating an oval window, sending pressure waves in the fluid and the cochlea, doing a basilar membrane, vibrating the little hair cells on the organ of Corti, right? So it's more, you have to take it deeper. Like the stuff we did in lab was more superficial. And in lecture, we talked about, but how exactly does it turn into an electrical signal? So if that helps.
Okay, thank you.
Other specific questions? Yeah, this is good. It also helps me because then it's so hard for me to like, where do I even start?
If there is like one topic that you are like, oh man, I'm going to write like the best question about this, and you're like, this is totally going to test my students, what might that be? Like I know like we've gone over so many details of all these different things, but if there's something that like maybe we've forgotten or we went over quickly or is so important.
Right, it's all important.
I know.
Yeah,
but I'm just trying to weed out that like one tricky question where that's going to like stop me for 15 minutes thinking about how am I going to write this free response.
And again, that's going to be different for different people. I mean, it's part of the reason on that last exam that I put that like write your own question one is because I knew sometimes people study really hard, but they might have studied something that wasn't on the exam and then they're like frustrated. So it's like, oh, show me what you know, give you a chance to like, but it really depends. Yeah, that's going to be different for different people and.
Totally.
Yeah. Professor, I would love to go over, I got confused this morning on the warm up around the the light coming in and hitting what you were just talking about. If we could just go through that again.
Sure. Let's see. Yes, I mean, the basic idea is you've got the rods. And like if we're in the dark, so we have our opsin, and it's got cis retinal, the retinal in the cis conformation. So we're still in the dark. We've got these sodium channels that are letting sodium in. And these are held open by cyclic GMP. Cyclic GMP is necessary to hold these sodium channels open. And so we're in the dark. Sodium channels are open. Sodium's coming in, which means the membrane voltage is going up, and it's around, what is it, minus 40 millivolts. So this is the situation that we're starting with before light hits the retina. And now we're going to, light arrives. So when the light comes in, all of a sudden the cisretinal turns to transretinal and detaches. All of a sudden my little opsin is activated. And it's going to trigger this G protein we're calling transducin. Transducin is just the names people have given this particular one. You can just call it a G protein. You can call it transducin. It's the same thing. That transducin is going to start a whole big second messenger pathway. That's going to create phosphodiesterase. Or create. It's going to activate phosphodiesterase, which we know does what? What's the job of phosphodiesterase?
Breaks down cGMP.
To break down.
Yep, it breaks down cyclic GMP, which means the sodium channels close. And now without that sodium current, the voltage is going to go back down to resting. The VM is going to go down to like minus 70 millivolts. And ka-ching, that was it. We have a voltage change due to the stimulus, right? That's what transduction is. There's, stimulus was light arrives. Transduction is it's creating an electrical signal. The electrical signal is the membrane voltage dropped from minus 40 to minus 70 in response to the arrival of light energy. And that is phototransduction in a nutshell.
That makes a lot of sense. So like in this case, similar to what we talked about before, when you introduced the second messenger, like the conformational changes, instead of it's like the, I forget what it's called, coming in and binding, it's the light that's doing that.
Right, exactly. Instead of a ligand binding a receptor, you have the light causing a conformational change, which triggers the second messenger system.
I see. Okay, thank you. That makes a lot more sense.
Cool. Right, and here, let me see if I can do this.
So in terms of once the VM goes down, then and at the neurotransmitter level, will there be a release of the glutamate?
The glutamate will slow down in response to the light. But that's not even as important, because mainly what I want you to get is that there is a electrical change that's happening in response to the light in the cell. And then lots of things happen downstream from that as we percolate through the retina. But the core here is there's an electrical change that's happening, and that is going to then cause a modulation of glutamate release, which is then going to cause lots of downstream stuff in the bipolar cells and horizontal cells and all that in the retina.
Yeah, because I was looking at it yesterday in the book, and it was a little confusing, like going below. I mean, from that point where the glutamate is.
Right, so don't even go there.
Okay.
Right,
Because it's like opposite.
It's opposite. Like, I said that it is a little counterintuitive. Normally you think we're exciting it, it's going to get higher, but it's still the signal, and all you need to do is invert it, and you still got the information you need. So it's just the way it is. So this is phototransduction in a nutshell. All right, what comes next? Any other questions about specifics?
Just a question. I missed what you said. So hyperopia and myopia, that was on the lab, but that's going to be on the test as well, the lecture exam?
No.
No. Okay.
No, that would be pure. This is, like I said, this is going to focus more on lecture material. You should know about the, flow of aqueous humor and glaucoma and things like that. You should know about the stuff I just mentioned about phototransduction in rods and cones, but it's not getting into things like visual acuity or anything like that.
Okay.
Beyond, I mean, you should know that the rods are on the periphery and low acuity, and the cones are in the middle center with high acuity.
Where does after the aqueous humor drains in the canals of Schlemm, where does it go?
Actually, it ends up back in the jugular vein along with the CSF.
Okay.
Yeah, no, a lot of the fluids in the inner ear, the fluids in the eye, and the CSF all ultimately all go down into the same sewer and back into your venous circulation. All right. So at the very beginning, kind of started, we were still talking about neurons even. When a neuron releases neurotransmitter and is getting bound to receptors, we want to make sure that the signal does not just keep going forever. How do we make sure? So the signal comes down the axon, neurotransmitter gets released, binds the receptor, but eventually there's no more signal coming down, and I don't want the signal to keep being here. I want to take the signal out and put it back to getting ready to wait for the next signal. How do I clear all of that neurotransmitter out of the synapse?
Reuptake or enzyme breaks down the neurotransmitter.
Yeah, so one way is the reuptake. You just kind of vacuum it back up into the presynaptic terminal. There's breakdown using enzymes. And actually it can be a mix even, like with acetylcholine, you have acetylcholine esterase to break down the acetylcholine, but then you actually reuptake the choline in order to reassemble and make more acetylcholine. And then what's the one other way we talked about?
Uses out.
They diffuse out.
Yeah, just kind of diffusing out into the intracellular space. Which of the glial cells was responsible for kind of maintaining the external milieu and making sure that things aren't floating around that shouldn't be, and that the ions that should be there are?
Astrocytes?
Yeah, the astrocytes. What else did astrocytes do that's super important?
Blood-brain barrier.
Yeah, the blood-brain barrier. Again, it's very difficult to cross from the bloodstream into the space around the cells in the brain because of this blood-brain barrier. Things are very tightly controlled. What kind of cells did we say that could make myelin sheaths?
Oligodendrocytes
Schwann
and
cells.
Schwann
It would
cells.
be oligodendrocytes in the CNS, Schwann cells if we were in the PNS. What else? We talked about the big picture. You should make sure you're aware of the big picture of what are the two, at the very top, what are the two branches of the nervous system?
CNS and PNS.
Yes, CNS and PNS. And then what would be the two main parts of the CNS?
Brain and spine cord.
Okay, brain and spinal cord. And we spent a lot of time looking at the brain, not so much about the spinal cord, but again, the spinal cord, we talked about a lot of it is just kind of carrying signals in and out to the brain, but then it also has some important integration with some reflexes as well. We'll come back. PNS, what were the two branches of the PNS?
Motor and sensory.
There was motor and sensory. And again, sensory, we talked about being there's both internal senses and external. You know, we spent most of our time getting into the details of the external, like hearing and vision and things. But as we continue on in the semester, we're going to see lots more internal sensing as well. Stretch of your stomach and intestines, chemoreceptors, like monitoring the contents of your digestive tract and your bloodstream. Receptors monitoring the stretch of your blood vessels and even your atria, you know, the chambers of your heart to make sure the blood pressure and everything is proper. So there's going to be a lot of internal sensing that you don't normally think about or you're not consciously aware of, and then lots of external stuff. You know, for each of the external senses, like I mentioned last class, make sure you understand the basics of the sensory transduction. You know, we talked about, again, how do your taste buds work? How do you actually, what does it mean? I licked the lollipop and it tasted sweet. Like you can say that. I mean, everybody knows that experience, but on a physiological level, what the heck does that mean?
Your taste buds are detecting glucose molecules or chemicals that then they can transmit up into the brain as an electrical signal.
And how is it, what's probably happening to actually allow them to know there's a glucose molecule there?
They have receptors that accept glucose and receptors that accept salt. And
right. Yeah, so there's going to be receptors that bind glucose as a ligand, and that's going to start some pathway that ultimately sends a message to the nervous system.
so the external senses are then like further categorized into like the general senses and the special senses?
Correct.
Okay.
Yeah, external are the general senses, which are all over your body, the touch, the temperature, the pain, and the special senses, which were the ones in the head, the hearing, the vision, the taste, olfaction.
So the the vision and hearing, are they considered as mechanical thing?
Are you
Vision
talking
and
vision? What is vision? What's the stimulus for vision?
it's light.
Light. So that's not mechanical. Mechanical is some physical force, right? Mechanoreceptors are when something actually physically moves or stretches. That's like hearing, there's an actual moving of your eardrum, vibrating. In touch, there's an actual stretch of your skin. The cells literally stretch. That's what opens the little channels, is the stretch of the membrane. So mechanoreceptors are some physical force that is being transformed into an electrical signal. Whereas in vision, light energy is what is being converted into an electrical signal. So does that make sense? I can't hear you.
So hearing, it's not considered.
But you just muted yourself.
So the hearing, it's not considered a mechanical.
No, I said it is.
It is, okay.
Right? Because it's about the motion of your eardrum. The air vibrations. Your voice are these compression waves through the air, which then wiggle and jiggle your eardrum back and forth, which ultimately wiggles and jiggles the hair cells back and forth. And that movement of the hair cells is what is getting converted into an electrical signal. So hearing is actually ultimately a mechanoreceptor.
I have a little side tangent before I forget. We talked about, like, kind of back to the CNS, we talked about like pyramidal tracks,
Right.
reticular activation pathways, and the limbic system. They kind of seem like little one-off things that also go involved in the nervous system. Would you like categorize those in a way, or kind of?
So two of them in particular. So limbic system.
Mhm.
Yeah, and the reason we call it system was because it's not a specific region, like most of the other stuff we've been talking about is a particular region. Limbic system were a variety of interconnected areas all involved in emotion. So that's why we give them the name. And it included like the hypothalamus, the hippocampus, the anterior cingulate, the what else? The insula, things like that. Reticular activating system. Also, not a specific region. It's a collection of regions and neurons within the brainstem that send messages up to the higher brain regions that are necessary to maintain consciousness. And this, particularly important, like if this is not functioning, you're in a coma. So people talk about this, in a clinical setting, because this is critical for actually being able to, be conscious.
So it's like the Glasgow Coma Scale, where it's like they respond to stimuli or like painful stimuli versus just moving things on their own and stuff like that. Maybe.
I think if you're in a coma, I'm not even sure how much you're going to respond.
No, but it's like a scale. So like, I mean, it depends like how conscious you are, right?
Yeah. So it would be in general, if someone's in a coma, you can't wake them up.
Right. Okay.
Right. And then when I talked about the extrapyramidal pathways, that's not a system. That is something else. I was talking about the vernacular people use when they're talking about, we talked about in the precentral gyrus is where you have your primary motor cortex and those neurons that are all coming down to operate your skeletal muscles from there. We call those the pyramidal tracts. But then I also mentioned that there are other pathways involved in motor control, like those deeper, the basal nuclei and stuff. And those are what I was talking about, extrapyramidal, like parts of the motor control that weren't specifically this connection from the motor cortex.
Okay.
So both of them go through the decussation area spot?
The pyramids are the ones that decussate specifically, and they call the decussation of the pyramids. Everything decussates at some point.
But not the extra, not the one that's coming from.
Everything, all the muscles that go down to your body, or all the signals that go down to your muscles at some point cross over, but in different places. Right. What else? Other things in here.
Sorry, just to kind of go a little bit further into the extrapyramidal tract. So like neurons from like the cerebellum and
I wouldn't say the reason I brought it up in the first place, I didn't mean to get confusing. It's when you are reading the side effects, particularly for antipsychotic drugs, they often talk about extrapyramidal symptoms.
Oh, okay.
And when you see that word, I wanted to give you some context for what the heck does that mean. Again, I talked about the tardive dyskinesias. Kind of late onset problems with movement. but it's a different thing than with the like paralysis that you'd get from a stroke that damages the direct control of your motor. These are going to be more, again, you can't stop moving, kind of similar like Parkinson's, right? Parkinson's is also dysregulation of these deeper gray matters involved with motor modulation.
Okay.
So that's, but I didn't mean it for it to be a big thing. That was more of an aside. Probably shouldn't even be spending that much time about this right now on our review.
No, I just wanted it like in relation to all the other notes I have from that lecture, just like just categorizing the systems of limbic and reticular activating systems. And so this is something so different.
Right. So these systems are very important to know about. You know, you will very likely have questions on one or both of these because they are kind of at the core of how we describe important parts of the brain that are not in one place, but kind of more collectives that have a common function. Whereas that other stuff I was saying is more about terminology that's used when you're trying to describe side effects of a drug.
Gotcha. Thank you.
All right. Other questions or comments, or other questions, places you'd like me to go before I kind of continue? Okay. Oh, and I also need to get my little clock thing going again. So, man. This is, I think there's something weird. Like maybe every time the iOS got updated recently, and now like all my iOS stuff is just collectively acting weird and all dimming together in ways that don't make any sense. So I apologize for a moment. I can't even see what I'm doing here. Okay. Okay, now I can. And hopefully, I don't know if you hear, I've been trying to turn off all my messaging. I put everything on airplane mode, and yet I keep, on my side at least, I just keep hearing these beeps and stuff. I hope
you were blowing up the entire lecture.
Oh, damn it. Yeah, I put.
But it's coming through Wi-Fi, right? So you're receiving all those messages through Wi-Fi, which is on in airplane mode.
Oh, so that's
Dr. E, we know you're really popular at this point, loud and clear.
I mean,
right.
It's more about we're all like, it's all about the election right now. This isn't about popularity. This is about everybody freaking out. Yeah, let's see. I apologize for this. I need to put, at this point, it's too late. On next class, I will go in not just airplane mode, but turn off Wi-Fi.
Well, then only on those devices, because then you won't be able to do the exam. So just to clarify, like.
Right. All right. So back to this. Motor, sensory. So limbic systems, these are, you should know. Let's erase those from here so we can continue with where I was at. Dagnabbit. Yeah, I apologize, because that's something I know is distracting for me, and I'm sure it's been distracting for you. So that's too bad. All right, motor. Motor is then further subdivided, which you should know. What are the subdivisions of the motor, or subdivision of the PNS, subdivision of the nervous system?
Somatic and
somatic,
autonomic.
Again, somatic, which is the one that operates all the skeletal muscle we've been talking about. This is the one that has the neuromuscular junctions, releasing acetylcholine at nicotinic receptors at the neuromuscular junction on skeletal muscles to make them twitch. If you keep sending messages down, you go into tetanus, blah, blah, blah. So this is the tie-in to everything we've been talking about today about skeletal muscle. Autonomic is further subdivided.
Sympathetic and parasympathetic.
Sympathetic, parasympathetic. And again, sympathetic, the fight or flight. Again, speed up your heart versus parasympathetic, slow down your heart. Sympathetic, dilate your pupils. Parasympathetic, constrict your pupils. You know, in general, as a rule, sympathetic is getting ready to deal with some, emergency or stressor. Send the blood to your muscles and suppress digestive processes, because we don't got to worry about digesting our last meal if we were about to get eviscerated by a lion. We need to put our resources in staying alive. Parasympathetic, on the other hand, is the rest and digest. It's about promoting homeostasis and healing. It's like, let us stimulate the digestive processes and get this nutrients into our body here and slow down the heart and kind of just take care of things. Again, also constrict the pupils, things like that. And again, like I mentioned, in general, they tend to push and pull against each other, right? As the light gets a little brighter or a little dimmer, one or another of these is pushing and pulling to make your pupils a little bigger or smaller. So it's not like they are in constant, you're constantly freaking out or constantly kind of lying there. What neurotransmitter is released by the second neurons in the parasympathetic nervous system?
In parasympathetic, it'll be acetylcholine, and then in sympathetic, it'll be epinephrine.
Yeah, epinephrine or norepinephrine. So norepinephrine is more the neurotransmitter that's released. Epinephrine is when it's more of a global broadcast into the bloodstream as a hormone.
One question, Professor. The adrenergic norepinephrine and epinephrine, they're always excitatory?
No.
They could be both.
Yeah, exactly.
Okay.
Mm-hmm.
Sorry, can you go over that again?
Yes, she was asking about, because both of the receptors here are second messenger mediated, whether it's the muscarinic cholinergic receptors on the targets. Or here we have the alpha and the beta adrenergic receptors. These are both second messenger mediated, which means you don't know what the effect on the target's going to be unless you look at that specific target. Right? Somatic, we talked about, always excitatory. If you send a message down a somatic motor neuron, the muscle is going to contract. Whereas over here, sympathetic, you might speed up the heart, but you suppress the digestion, right? So it might excite or it might inhibit depending on the particular part of the system you're looking at. So you can't make any, blanket assumptions about excitatory or inhibitory.
So, but that's looking at the effect where you can't tell if it's excitatory or inhibitory, but like if you look at each individual thing, like norepinephrine and epinephrine, they always cause an excitatory, or you still just cannot tell?
Don't go there.
Okay, done, fine.
Right, because all we can say is they bind a receptor, but then there's a whole second messenger pathway, and who knows what happens after that.
Okay, there we go.
So what else? And then I talked about, there's a few things that are purely, what things are purely sympathetic?
Blood vessels.
Sweat gland.
Adrenal glands.
Glands.
Well, sweat glands in particular. And actually, I should say, yeah, adrenal gland, the adrenal medulla, and the most of the blood vessel muscles as well. But for the most part, they tend to push and pull against each other. You know, you should, I'll probably have some question kind of like we had in the warm-up. Like if I wanted to put a drop in your eye that made sure your pupil did not constrict when I shined a light in there to doing an eye exam, what kind of drug would I use? Again, basically what you need to do is figure out which division.
A cholinergic.
Or say it again.
Cholinergic antagonist.
It would be a cholinergic antagonist. Cholinergic because it's parasympathetic to constrict the eye, and I want to block it, so I use an antagonist. If I wanted to, like for blood pressure, I want to keep the heart from speeding up, it would be an adrenergic antagonist because I want to inhibit the effect of the sympathetic nervous system. If I want to stimulate, if I want to dilate the bronchial passages because someone's having an asthma attack, I want to actually activate sympathetic, then I'd want an adrenergic agonist, right? So you should be able to answer a question like that because there's a lot of drugs that are out there and that are used a lot that work by either being agonists or antagonists to either acetylcholine or epinephrine and norepinephrine, and you should kind of understand the basic idea of why that is such a useful thing. Blah, 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 blah. Brain. So then we can go to the brain. You know, we talked about the big picture stuff about the flow of cerebrospinal fluid. You should first know the basic regions and kind of how they connect. You know, the forebrain, you know, which created the cerebrum and the diencephalon. And then the midbrain, that's the midbrain, you know, the superior inferior colliculi and the substantia nigra. The pons and the medulla, which we kind of collectively just threw together as all these autonomic functions like cardiovascular control, respiratory control, you know, vomiting centers, sneezing and coughing, things like that. And then we threw on the back was the cerebellum, which did a lot of that kind of fine motor coordination. So you should have the bigger picture. Then we spent a lot of time in the cerebrum. You should know about prefrontal areas with executive control, you know, precentral gyrus frontal areas for motor, you know, somatosensory and the parietal of the postcentral gyrus. There was the, what do we have back here?
Primary visual cortex.
Primary visual cortex. And then we had the auditory cortex up in the temporal lobe. And there was lots more, but again, I think that's good enough for now. We talked about the deeper gray matter, which is involved with motor control as well as the amygdala. What did the amygdala do?
Like fear and anxiety.
Yeah, fear and anxiety. And then another piece of deeper gray matter, the hippocampus, which was down here in the floor of the temporal lobe. What did the hippocampus do?
Like emotions and memories.
Yeah, it's got motions and memories. So it's part of the limbic system, but then it's very important to understand the idea of consolidation and reconsolidation. What does the idea of consolidation mean?
Short-term to long-term memories.
So that idea that when you first learn something or memorize something or experience something, it's in this kind of temporary version. Like if you take if you put a voltage across your brain, you're going to lose it. But an hour later, it will be in a more permanent way that is going to stay as a memory. So there's this conversion of this temporary form of the memory into this long-term form that's consolidation. And we talked about reconsolidation, which I think is worth knowing about because it makes you have a little more humbleness about, I'm sure I remember it that way or something, and realizing just the more we understand how memory works is you can't totally trust your memory as much as you wish you could.
So consolidation just refers to the first time you have learned it and like it's stuck, and then reconsolidation is every time you recall it and form new memory and kind of adjust.
Yeah, exactly.
Okay.
You kind of put it back, but it's been contaminated by whatever new stuff it got mixed with when it was back in short-term memory.
Okay. And yeah, like with older people, it's because they have an issue with that like protein synthesis or something where when they do when you form the long-term memories or new ones, I thought it was like a physical problem.
It is. Well, no, but I was just saying in these studies, the way they've learned about this is by using drugs that block protein synthesis. I don't know how that works in terms of humans or human aging or anything. So
oh, okay.
yeah, that is not something I meant to say if I said that. We also talked about different kinds of memories or learning. What are different kinds of memories or learning?
There's
Declarative and
procedural. Declarative, also known as explicit. That's where you know, like, I know that the hippocampus is involved in consolidation, and that consolidation is the transition of short-term to long-term memories. You know, so that's declarative. Then we also talked about procedural or implicit learning, where you are learning, you're recognizing patterns that you can then use to influence behavior, but you don't even know that you are actually doing that. You more just kind of, it's working kind of under the radar. But experimentally, you can show that you have learned something and that you are performing above chance at these different tasks. So your nervous system, your brain has observed stuff, learned stuff that's influencing your behavior, even though you don't have a conscious perception that that is true. Again, the people who do these studies, they feel like they're just guessing. But in reality, they are performing above chance, so they're not just guessing. So it's officially 12:35.